Because native revegetation is a business, native seed producers harvest seeds of several species from single-species production fields. Additionally, many species that are commercially available are harvested from wild populations. In the Midwestern U.S., only about 10% of the native plant species are commercially available. For the rest, you'll need to harvest them yourself. Remnant natural areas are often utilized for wild harvested seed. Some ecosystems like prairies, savannas, and wet meadows are no longer widespread, so these remnants are of high conservation value. Over-harvesting seeds from remnants can threaten the persistence of native plant populations. Without seeds, new generations of plants are not produced. The risk of over-harvesting is especially high on small, high-quality remnants with public access. Some guidelines to follow when collecting seed. Avoid repeated annual harvests of the same remnant areas. Leave at least half of the ripe seed on the remnant plot. Species on federal and state endangered or threatened lists cannot be collected without proper permits. Permits for seed harvest are required for public lands and those owned by conservation organizations such as the Nature Conservancy. Management such as burning that promotes seed production should not be repeated annually or applied across entire sites. Manipulation of a remnant prairie to maximize seed production, such as repeated annual burns on the entire site, herbicide treatments, or fertilization, is inappropriate. Even burning should be limited to only a portion of a remnant in any given year, and each portion should be burned on a rotation and at different times of the year at varying intervals. When harvesting seeds from wild populations, it is important to maintain the integrity of the natural plant community, as well as collect seeds in a way that will benefit your restoration in the future. A plant population that is genetically diverse is more likely to survive extreme environmental conditions or disease. If the population has a diverse genetic makeup, then some members of the population may have traits that allow them to survive extreme conditions that might arise. The way you collect seed can affect seed genetic diversity. When harvesting seeds from wild populations, here are a few rules of thumb. First, generally speaking, near neighbors are more closely related genetically than distant individuals, so it is important to collect seed from throughout the population. Second, collect equal amounts of seed from multiple individuals in the population. Do not collect most of your seed from few individuals. Third, if possible, collect from multiple sites within the vicinity of your site and use approximately equal amounts from each site in your seed mix. Fourth, harvest seed over time to avoid collecting seed only from early ripening populations. In addition to using best practices to maintain the genetic diversity of wild harvested seeds, making sure seeds used for restoration are well suited to the local conditions of the restoration site is important. Many Minnesota species occur across broad geographic areas and experience a wide range of environmental conditions. For example, wild rose is found across the North American Great Plains, so populations must persist through very different local stresses. Choosing seeds from different regions and environmental conditions can have significant ramifications. Across this range, the stresses plants experience due to temperature, precipitation, disturbance events, soil conditions, pathogens, and predators vary greatly. Genetic differences have evolved in response to these stresses, and populations well suited to one set of environmental stresses may not be well equipped to thrive under another set of environmental stresses. Let's look at an example of how choosing a seed source of wild rose from either southwestern Minnesota or central Kansas can make a significant difference for a restoration site in central Minnesota. A wild rose population of a Minnesota prairie differs from wild rose populations elsewhere in the U.S. as it has evolved to withstand extremely cold winters, with little or no snow serving as insulation through the coldest months. Only the individual plants genetically adapted to survive extremely cold winters will survive to set seed. When plants that tolerate local conditions best produce the most seed, they begin to make up an even greater proportion of the population. Seeds from the plant genetically adapted to survive the local conditions will establish across the landscape. Only young plants from the plant able to withstand the extreme cold mature into adult plants and are able to produce more offspring. Over time, the genetic makeup of the population will shift if the plant's response to these conditions is inherited from one generation to the next, and for plants that thrive in Minnesota, this means the local population is adapted to future cold events. However, remember that local conditions are not just climatic conditions, such as fluctuations in temperatures, but can include tolerances to wet conditions, saline soils, etc. It is not easy to know which conditions a population is genetically adapted to, and whether this could prove to be an advantage elsewhere. Over thousands of years, populations have experienced many different episodes of varying environmental stresses which have influenced a population's genetic legacy. Many restorationists play it safe and use seed sources as close to the project site as possible. They're assuming these sources have a home site advantage and will be better performers over the long haul. Remembering our example of sourcing wild rose seed from either southwestern Minnesota or central Kansas, the seed sourced from Minnesota is likely to produce a population that is better genetically adapted to local environmental conditions of extreme cold events. A population that establishes from Kansas seed is likely to produce many offspring that will not survive extreme cold events, and the population on the restoration site will struggle. In addition, the seeds of some species readily disperse through the landscape. These immigrants contribute to the genetic makeup of the local population. Complicating things further, how well matched seeds from one location are to another will not necessarily be apparent for many years and may only be apparent for a few traits. For example, poorly adapted individuals will not be evident until a disease outbreak points out the lack of resistance to a pathogen, or frigid conditions cause physiological damage to less hardy individuals. There are three basic approaches for matching sources to local conditions at a revegetation site. Select seed sources that are close enough to have likely experienced many of the same conditions over time. How far away from a restoration can seeds and plants be collected without compromising revegetation outcome? Distances recommended range from 20 to 200 miles. These distances often correspond to differences in climate. Geographic patterns of genetic variation have only been explored for a few species. Match seed sources based on a broader array of environmental conditions such as soils, topography, and local climate. This approach for sourcing seeds is likely more feasible when wild harvesting seed for a project of known conditions rather than using seeds purchased from vendors. Delineate seed transfer zones. Seed collected within a particular zone is recommended for use anywhere within the mapped area, but not beyond zoned boundaries. These zones correspond to regional differences in climate and landform. They offer some of the advantages of the other two approaches, although project sites near zone boundaries could end up using seed from much further away than is necessary or optimal. Some seed source guidelines use a combination of these, with different approaches being recommended for different situations. The Minnesota Board of Water and Soil Resources has seed source matching guidelines, and the article from Broadhurst further discusses the different strategies mentioned here. For some applications other than restoration, uniformity of traits, rather than genetic diversity, is valuable. To promote desirable uniformity, specific individuals are chosen from trial plantings to become the parents of an entire seed production field. This process is called artificial selection. The plants produced from this process are called unimproved cultivars. Artificial selection is common for improving plants used in agriculture or horticulture, but is controversial in ecological restoration for the following reasons. First, in general, seed of native cultivars often has lower genetic variability than wild material. This will lead to decreased ability to adapt to changing and extreme environmental conditions. Second, traits selected for agricultural and horticultural purposes are not necessarily adaptive for natural communities. And third, cultivars are likely to set more seed and establish more readily than plants from wild populations, since those traits are required for commercial production. This might become problematic for your restoration, as the cultivars may act like invasive plants, crowding out other desired species and leading to low species diversity. Additionally, if native cultivars are planted on a restoration site and spread into nearby remnant populations, locally adapted genes can become rare, a process called genetic swamping.